Okay, so welcome to this next video in which we're discussing the effect of sympathetic stimulation on the heart. Okay, so we want to now see how protein kinase A of the first type, i.e. the free type that's not associated with an A kinase anchoring protein, is going to alter, it's going to phosphorylate troponin, and it's going to alter the sensitivity of troponin to calcium. So at the moment we're just having a reminder of the contractile structures and how we control the commencing of the contraction of a sarcomere. So I've just told you about tropomyosin, which is this protein which is wrapped around the actin filament here. So this is this sort of spring-like structure that I've drawn wrapped around the actin filament. And basically, when the heart is at rest, what tropomyosin is doing is stopping the myosin filaments from being able to interact with the actin filaments, and therefore stopping the myosin filament from pulling the actin filament this way in this case, and this way in this side's case. Okay, right. Now, the way in which calcium causes the commencement of contraction is that the calcium binds to a protein that is controlling the position of the tropomyosin on the actin filament. So, basically, let me show this. If we have our actin filament here, so this is our actin filament. Okay, so these are these actin proteins all polymerized together to form a long actin strand, and I will show them in green as we've previously used that convention. So, the tropomyosin winds around this actin filament and is held in place by another protein called troponin, yes. Okay, so here is our tropomyosin wrapping around. So we'll have this drawn in blue here. Actually, no, not blue, because we've been using blue to denote the myosin filament. So here's our tropomyosin in turquoise. Okay, right. Now troponin is actually a complex of 3 different proteins. So I'll draw these here. You have 3 separate proteins bound together, and these 3 blobs that I've drawn here, this is the troponin protein complex. So it's many proteins all bound in a single complex. Okay, now the one that is closest to the actin filament, the one that is bound to the actin filament, this is known as troponin I. Okay, so this is troponin I. And this is the one that's involved in holding on to the actin filament, basically. So troponin I is in orange here. Now, the middle protein here, this is troponin C. So let me write its name out, troponin C. And we'll colour it in a pink colour. So troponin C is the middle one of this troponin complex, which is in pink here. And then finally, this one up here, this is troponin T, the one that is bound to tropomyosin. Okay, and their names basically reflect what they do, at least in the case of troponin C and troponin T. Troponin T is called troponin T because it interacts with the tropomyosin here. Troponin C is called troponin C because it binds to the calcium. Right, now usually in the resting heart, the troponin I is positioned correctly and is holding the tropomyosin in the correct position, that it's over the myosin binding sites on the actin monomers, and it's stopping the myosin heads from being able to interact with the actin filament. However, when calcium goes up in the cell, calcium will bind to troponin C, and troponin C will cause a change in conformation in troponin I, so that now the whole thing moves, and the tropomyosin here in turquoise is moved out of the way, basically, so that it no longer covers up the myosin binding sites on the actin monomers. So this is tropomyosin. Right. So what is protein kinase A going to do? Well, basically, the type 1 protein kinase A, which is the free soluble type that is not bound to an A kinase anchoring protein, it's going to be activated by cyclic AMP in response to noradrenaline binding to the beta-1 adrenergic receptor. And this protein kinase A catalytic subunit is then going to be released. So we'll show this as a large dot in vivid purple. So this is the catalytic subunit of protein kinase A of the first type. And this is going to go in and phosphorylate the troponin I. So it's going to go in and interact with troponin I and phosphorylate that troponin I. And when it does that, it's going to increase the sensitivity of the entire troponin complex to calcium, so that calcium will bind to the troponin C, and the effect that it has, the efficacy of it, will be greater, basically. So it's more likely to induce a conformational change in the protein that will then lead to the tropomyosin being moved out of the way of the myosin head. So it's going to increase the effect of calcium on these sarcomeres, so that you get increased contraction in the sarcomeres in a single sarcomere for a certain amount of calcium. So the two ways now that we have increased the ionotropic effect of a cardiomyocyte are that we've increased the size of the calcium spikes so that we can hopefully recruit more sarcomeres and make them more contract, therefore we'll get a greater force of contraction. And also we've increased the sensitivity of an individual sarcomere so that, again, that sarcomere will contract with greater force. We increase the force that each sarcomere is producing and also increase the number of sarcomeres that we're recruiting. Okay, right. So now what I want to discuss is one last little thing, which is the fact that the cyclic AMP level within the cell does not merely go like this. From what I've told you so far, you might think that cyclic AMP, if we plotted cyclic AMP concentration, it would look something like this. i.e., if we plot it against time, then here's cyclic AMP concentration maybe before we start stimulating with the beta-1 adrenergic receptor, and then we apply the beta-1 adrenergic receptor, it goes up and it remains at some high level continuously. That is not what you find in cardiomyocytes. Instead, what you find is that when you apply the beta-1 adrenergic receptor, so here's when you apply the beta-1 adrenergic receptor, what you get is you get a spike, then you get an interval, and then you get another spike, and it continues on in this way. You get these oscillations in cyclic AMP level over time, just like you have oscillations in the calcium concentration over time. And in fact, the oscillation in calcium concentration is responsible for this oscillation in cyclic AMP level. So let me explain why this is. Okay, right. So we have seen then, if I get my pages right, where are they? Okay, where have I put them all? Not that. Let me just find this piece of paper. Okay, never mind, I can't find the piece of paper. Maybe it's over the other side here. No, this isn't what I want. Where is the piece of paper where I drew the initial pathway on? Ah, here we go. Right. So, we've seen that the noradrenaline binds to the beta-1 adrenergic receptor and activates the GS, heterotrimeric G protein. Okay? This then goes down and activates these adenylyl cyclases, okay, which start producing cyclic AMP. The cyclic AMP then goes on to activate the protein kinase A. However, these adenylyl cyclases, adenylyl cyclase 5 and 6, they are actually inhibited by calcium. Okay, so if I get another piece of paper, if I draw it down here, we know what the calcium concentration inside the cell is doing. We know that calcium oscillates within the cell. So we know that if we plot calcium concentration versus time, so here's calcium on the y-axis, and here's time on the x-axis, what we get is that calcium goes up and then reaches a peak where the calcium starts to inhibit the type 2 ranitidine receptor, and also the action potential is finished, so the L-type voltage-gated calcium channel is no longer allowing calcium in. And then what happens is the circa 2 pump returns that calcium into the sarcoplasmic reticulum, and it goes down again. You then have an interval, another action potential comes along, the L-type voltage-gated calcium channel releases calcium onto the type 2 ranitidine receptor, which opens and releases calcium into the cytoplasm. So calcium goes back up, and then the same thing happens again. Now, basically, when calcium is very high, what that will be doing is it will inhibit these adenylyl cyclase 5 and adenylyl cyclase 6 enzymes. And the reason it inhibits them is because there is a magnesium ion at the center of the active site of these adenylyl cyclase enzymes. Okay, and we'll continue this discussion in the next video.